Welcome back to the free webinar on Omicslogic Data Science. In today's session, our team will be sharing information, examples, and an overview of the Omicslogic training resources designed for anyone to learn data science for biomedical data as seen in application to projects sourced from current topics in biomedical research. Over the course of the past several years, our team has been collaborating with a number of academic institutions around the world that have participated in content evaluation, curriculum review, and project design. As a result, the Omicslogic programs have been thoroughly evaluated and are known to lead to excellent outcomes. Importantly, the programs are designed to support students with minimal bioinformatics background, including biology and biomedical studies majors, as well as faculty interested in bioinformatics, data science, and biomedical research. The goal of the Omicslogic programs are to provide training in bioinformatics, enabling independent research guided by mentors and peer example, developing a growing community with shared passion for data-driven research and appreciation of citizen science, enabling students, clinicians, and faculty of all backgrounds to develop novel and independent research using latest technology in life sciences and biosciences. The program is designed to offer an introduction to statistical analysis and machine learning, and a broad overview of technologies and project examples to learn about and develop practical data science skills using user-friendly cloud technologies as well as R and Python coding. Omicslogic is a program for bioinformatics training that was developed in collaboration between Pine Biotech and the Tauber Bioinformatics Research Center. What we will discuss today is a result of hard work by an amazing team led by company co-founders Dr. Alfred Tauber and Dr. Leonid Brodsky. Dr. Alfred Tauber comes from a medical background and has extensive expertise in oncology, immunology, and biochemistry of inflammation, and has been recognized internationally for his generous support of many research projects and initiatives that support data science training and applications in medicine. Dr. Leonid Brodsky is the director of the Tauber Bioinformatics Research Center at University of Haifa and an expert in bioinformatics and biostatistics. He has numerous publications in mathematics and bioinformatics, and over the course of his career has developed multiple novel algorithmic approaches to biomedical data analysis. The rest of the team you see here includes experts in bioinformatics, computer science, and training. Some of our team will be presenting today. So let me introduce you to the mentors and the speakers of today's session. We have our CEO and co-founder, Ilya Brodsky. In his current role, he has extensive experience working together with industry and academic partners that are applying multi-omics integration and bioinformatics to a variety of biotechnology and clinical applications. Julia Panoff is a bioinformatician and has developed many of the resources we use for the Omicslogic programs. Dr. Harpreet Kaur is our Omicslogic trainer and expert bioinformatician who will guide the sessions and assist participants with projects and analysis during the program. Her area of specialization is cancer genomics and machine learning. Dr. Mohit Mazumdar is our Omicslogic project mentor and an expert in big data and computational biology. To start today's session, I would like to invite Ilya to speak now. Thank you, Ilya, and over to you.
Thank you very much, Pips.
Sorry, Ilya must have some technical issue. So he'll be joining in a minute, as I see.
Maybe Diksha, by that time you can tell them about the attendance certificate people are actually sharing. So
Uh,
yeah,
Sorry about that. Disconnected. Let me get back to my presentation. Okay, so welcome everyone to this webinar, and I'm really excited to be able to share with you a couple of things about this upcoming program. Now, I have to mention the reason why we are doing these webinars. The reason we're doing these webinars is because we know that there's an increasing interest in bioinformatics and in data science. There's a variety of people from different backgrounds, and so we're hoping that we can explore how we can adapt the elements that we have designed and developed for the Omicslogic programs to assist you at your level. Now, we're going to introduce to you some of the topics that we will cover in the program. We will give you an example from cancer research presented by Dr. Harpreet Kaur. And we'll also talk about some of the general challenges as well as opportunities for those of you that are interested in data science, how to develop skills that are needed and how to make sure that you understand the logic of the analysis, which is the main objective of these Omicslogic programs. This is not a workshop where you will get hands-on experience and develop skills. The reason for that is because we have only limited time, and really we are exploring how to best allow most of you to join one of the upcoming programs or benefit from some of the resources that we have available online. So for those of you that have joined to receive a certificate, unfortunately, this particular webinar will not come with a certificate. It's a free informational webinar about an upcoming program. For those of you that feel that the content of this webinar is not informative, that is okay. This is intended to introduce you to the upcoming program that we will cover. But we do hope to learn more about you and to really take away the feedback from the type of people that join these programs and join these webinars to make sure that we can be more effective at what we do. So again, thank you for joining. And I wanted to start by asking you a couple of questions, both about your current level education. And if you mention your education, whether you're in high school, you have an undergraduate degree, graduate degree, or postgraduate degree, if you would like, please share with us also what institution do you currently work for or study at, be it a school, a university, or a company. We would love to know, and we will save it in our records so that we know where people are primarily coming from. Now, for those of you that are coming from an international institution, we obviously are encouraging you to continue and attend these webinars, but we also have specific programs that are designed for different time zones and for different localities. So that's one of the reasons why we are interested to understand more where you're coming from. So to answer the questions that we will have throughout the webinar, we will ask you to just put in a number. So number 1 would be high school, number 2, undergraduate degree, number 3, graduate degree, and number 4, postgraduate degree. So please put your answer in the chat. Excellent. Thank you very much for sharing this information. Now, the reason why we're asking is because we know from previous programs that about 75% of all of our participants have either a master's or a PhD in some area of life sciences. And that is why a lot of the materials that we have are focused on that audience that either has a graduate degree or a postgraduate position, or they intend to pursue that career path. So you're genuinely interested in research problems, you understand how data is becoming a major part of answering those research questions, and one of those reasons drives you to pursue more experience with bioinformatics and gain more data science skills. So that's why I come to my second question. The second question is, what do you expect from this program? So thank you for everyone that answered. Unfortunately, if you did not answer, that is fine. We'll actually guide you to a free course where you can fill out a full survey about your background. But my second question is about, what do you expect to gain from joining this upcoming program? So this webinar, as I mentioned, is informational, but we're really wondering what might be the outcome that you are most interested in. So here are the options. Number one is to learn about omics data analysis. Number two, to get familiarized with data science and data science skills. Number three, an overview of statistical and machine learning methods. Or number four, apply data science to a research project. Okay, or five, all of the above. All right, excellent. I think this is very exciting for me to learn also, and this confirms what we are striving to do with these Omicslogic programs. We are designing these programs to help you both learn and apply these skills to a research problem. So today, we will talk about some of the skills, talk about some of the resources that we have to help you develop those skills. And then we will also talk about an example project that Dr. Harpreet Kaur worked in her research, which has to do with genomics data in cancer, and discuss how similar types of projects you will be able to develop during this upcoming program. So first, let's talk about bioinformatics and data science. And I know that there has been a discussion in the online community about both similarities and differences between the two. So as you know, bioinformatics itself has these three components: statistics and maths, computer sciences, and biology. And so when we speak about data science, a natural question is, why are we now speaking about data science as something new and not continuing to just speak about bioinformatics? And so let me briefly speak about what is typically expected from a bioinformatician. So a bioinformatician is not expected to be an expert in everything, but they should have some practical experience. And that practical experience is shown in a project. But here are some general skills that one might expect from a bioinformatician working in a research lab. They should have domain-specific data processing and quality control, general data transformation and filtering, applied statistics and machine learning. Applied means that you don't necessarily understand the theoretical components, but you know how to apply them efficiently. General data visualization and reporting tools, as well as very domain-specific data visualization. Specific data integration, so you know how to work in an environment where you might have to use APIs, databases, and specific types of files that have been compressed. And being able to write code with version control for data analysis tasks. The code should be reusable, efficient, and documented. And many people in the lab are required to write Python or R packages for reuse. So we will typically develop a script or develop some kind of a function that other people need to be able to use. And finally, an important component of this is being able to communicate data-driven insights or stories in publications, posters, and presentations, including multiple interactions with people that have minimal data science or computer science background that should still understand the insights that we're bringing to them. Now, how is this different from data science itself? Data science includes statistics and maths, computer science, and also some kind of a specialization. So really, when we talk about data science, it is much more agnostic of the domain that it is applied to. So according to such course platforms like Udacity, the essential skills of a data scientist are programming, machine learning, statistics, data wrangling, data visualization and communication, data intuition, and other related skills. So really, the components of data science and the components of bioinformatics are very much alike. However, when you think about the transferable skills, the skills that you're gaining out of this course, it is important to think of it as a building block for your career path or as a building block in your research. So one of the benefits in learning data science apart from bioinformatics is that you really gain a wider, broader perspective on how to apply data processing, data wrangling, visualization, and analysis skills to a variety of data sets that come your way. When we speak about bioinformatics, oftentimes bioinformaticians are going to have a very specific niche focus that prevents them from being able to deal with new problems effectively. And so this program is going to be able to provide you with a broader overview of some general skills that will go beyond your typical bioinformatics training. Now, some of these skills are going to be very important also in application to biomedical research and biological research, which does fall under the domain of bioinformatics, so you will see a lot of overlap. Now, what are some of the data challenges and opportunities that are happening in biomedical research as we speak? Over the past 20 years, there has been a phenomenal growth in adoption of high throughput technologies like next generation sequencing, which is both more and more affordable and widely adopted. In biotechnology, where people rely on insights and regular generation of data to drive experiments and validation, as well as in the pharmaceutical industry and clinical practice. These technologies produce an exponential growth of data that one has to deal with to get insights about a biological phenomenon or about a clinical application. Now, in parallel to this being driven by companies, a tremendous amount of data is publicly accessible, which means that many of us, as we learn about bioinformatics, as we learn about these data types, have access to data sets generated in professional labs that all of us can start analyzing. However, we're also facing a challenge. And the challenge is that this big data trend has produced a big data problem that precludes many biologists and clinicians from gleaning knowledge from these data regarding the nature of malignant processes and the relationships between clinical phenotypes and their molecular drivers. So this program will allow you to get some exposure to the nature of these big data sets and see how data science skills can help you address some of these challenges to make biological interpretation. Now, what do we mean by biological interpretation or biological insights that we glean from these raw data sets? So these data sets, like omics technologies, for example, next generation sequencing is a sequencing of DNA or RNA material found inside the cell. We can also talk about phenomics, which is the visual and phenotypic characterization of biological samples. And here we can see phenomics, for example, could be an image of cell lines. So when we look at the image and we can characterize the shape and the different properties of these cells, we can generate what's called phenotypic data. That phenotypic data is generated by looking at the outside of the biological sample. But omics data, from the molecular perspective, generates data about what's happening on the inside. For example, when we talk about DNA sequencing generates information about the differences in the DNA of a particular sample from a reference genome. And so here you can see an example of a table of somatic mutations that produces a characterization of different types of mutations relevant to cancer. Now, over the past several years, maybe 20 or so, there has been a growing interest in how the genomic information gets translated into functional proteins. And that goes through the process of epigenetic regulation. So this is information that is not stored in the DNA, but still affects gene expression. Finally, it has been known that you can easily study protein composition by looking at mRNA and microRNA expression. So we can capture RNA from the cell and we can study alternative splicing as well as level of expression of different genes. So together, all of these technologies provide a massive amount of information about what happens both on the outside and on the inside of the cell. As a result, giving us insight into how we can understand molecular drivers of conditions that might have clinical and basic mechanistic insights into the biology. Now, all of these kinds of data sets actually help us understand this data, but they first need to be processed. So when we process this data, we are generating different types of structured data from raw sequencing reads. And this is a major component of bioinformatics, is to understand the technologies that generate this kind of data and build pipelines that will process this data and turn them into structured tables. Once those data sets are structured, downstream analysis helps identify specific types of patterns that are associated with the different types of data that were generated. For example, we can talk about different types of comparison of conditions and finding variable patterns. We can talk about how those patterns could be mapped on the structural properties of different proteins and their interaction. We can talk about multivariate analysis of big data that allows us to summarize thousands of data points into a visual interpretation that might have association with phenotypic data, and the process goes on. So the mastery of these types of processing and downstream analysis methods pertains to bioinformatics specialization in these different types of omics data sets. Now, in the clinical or biomedical applications, these types of methods help us explore the relationship between clinical or phenotypic for clinical types of conditions data and omics data. And the major objective of such interaction and finding such association is to be able to improve the precision of diagnostic bioassays or biomarkers and the application of therapeutics. So design, discovery, repurposing, and assignment of specific treatment combinations to patients that have complex conditions. So a good example for that might be cancer, where a lot of times we don't have a clear target, or there's resistance to different types of treatments, or we need to detect cancer earlier. So we'll talk about an example in just a few minutes when Dr. Harpreet starts talking. So in the context of these different challenges, we can look at how to process, analyze, visualize, and interpret these different data sets to be able to generate a portfolio. So when you want to tell somebody, I know data science or I know bioinformatics, the way to explain to them that you know bioinformatics or you can apply data science is to demonstrate using a project data set. So for this upcoming program, we have prepared a number of curated data sets sourced from cutting edge research that is peer reviewed and comes from high quality publications and high quality labs, so that all of you can gain access to some of these examples and try to apply the same methods to diagnostics in cancer biology, which we'll talk about today, but also single cell and tissue composition, as well as the microbiome and infection. So you'll be able to actually study these different data sets and see how these data sets can be informative in the context of clinical application. Now, when we speak about the data science part of this process, we need to understand how the whole data science process works. So we need to talk about the experimental design and what factors or perhaps biases it can introduce into the data set. We talk about the process of cleaning, filtering, and preparing the data set for analysis, how to do exploratory data analysis when we use visualization and data mining to find patterns that might be informative and interesting, and oftentimes that generates a hypothesis. The hypothesis in a clear data science perspective is different from what we call a hypothesis in the biological sense. In the biological sense, a hypothesis is typically about some condition that has an observable pattern that could be validated through statistical analysis. When we talk about a data science hypothesis, we are talking about designing a model. Out of the many potential models that are out there, you have to understand what a model can produce, but also what factors need to be accounted for in each particular model. And so the design of the model that includes multiple statistical tests, and that can include machine learning models that need validation and that need a certain criteria for passing the validation, all of that falls into the domain of data science, being able to translate the data analysis into a model and then provide significant information about the performance of the model and the different outputs of that model to communicate with biologists and clinicians. And so a big part of what we'll do in this program is try to highlight some of these aspects and allow you to try them on your own so that you understand how this process works overall. Now, obviously, I mentioned machine learning and data mining, and so it would be useful to explain exactly what I mean by mentioning these terms. There has been an increasing interest in data mining, machine learning, classification, deep learning, and artificial intelligence. So I think from the perspective of a data scientist that works with biomedical research data, I think the utility of some of these models is tremendous. In particular, it's important to highlight that previously, when we relied on simple statistical models, oftentimes we were limited by the ability to deal with big data sets that contain a lot of variation that does not conform to the statistical assumptions about data which was generated before we see this emergence of high throughput data. So there's a lot of heterogeneity. There's a lot of features for each sample, and there are a lot of challenges in finding complex associations between feature to feature and feature to the object description or the phenotype. So what we'll do in this program is we'll take a look at some of these commonly used basic machine learning models like decision trees or like discriminant analysis, and see how computational methods that help with feature selection, that help with the robustness of models that we describe, and help us improve accuracy and the predictive power of these different models, has led to an emergence of this field of data science for machine learning. And so we'll talk about the different models and the different use cases for when these models and the selection of the appropriate model actually plays an important role in biomedical discovery. Now, it's not only important to know the statistical principles and to apply the models. In some sense, it has become easy. There are visual interfaces and simple packages that you can call a very complex model with a single line of code. But what's important is as you understand the variety of methods and different data types and can match the two to answer a question or to generate a hypothesis, you also need to make that model explainable. You need to communicate the results to your collaborators that come from the world of biology, biomedical sciences, clinical sciences, or even the world of business that includes business of research, where there's funding and there's different translational value to different projects, to explain what you have found in the data and why that is significant. And so we'll spend considerable time speaking about methods of visualization that are intuitive and informative for non-bioinformaticians or non-engineers. So to give you an example of how such projects are taking place today in the world of cancer biology and in the world of clinical application of bioinformatics, I wanted to introduce you to Dr. Harpreet Kaur. She is a trainer with the Omicslogic programs, and she will also share from her experience during the program when it starts in October. But today, I would like her to introduce some of her research background and explain how some of these methodologies have played a role in the research that she has done in her PhD. Please, Harpreet, you can take it over. Harpreet, are you there?
She says she's not able to unmute her.
Thank you, Ilya, for such a nice session. So my name is Harpreet Kaur, and I have a PhD in bioinformatics. My research is on biomarker discovery for diagnosis and prognosis of the liver cancer. So what inspired me to study liver cancer is the high mortality rate of this malignancy and a lack of precise biomarker that can help diagnose liver cancer at an early stage. It is sixth major cancer in terms of the incidences worldwide and fourth in terms of mortality. Liver cancer is oftentimes misdiagnosed or detected too late for any effective treatment. Another factor that contributes to poor outcomes for liver cancer patients is the frequent reoccurrence of the tumor. So with these concerns in mind, I have framed a number of research objectives with my collaborators to address these issues using bioinformatics. For the first project of this research, we use large-scale transcriptomics data to identify a diagnostic biomarker panel for the hepatocellular carcinoma, which is a major primary malignancy of the liver cancer. So in this study, we employed 30 datasets that containing nearly 4,000 samples to elucidate transcriptomics biomarker panel for the diagnosis and the prognosis of liver cancer, implementing various statistical and bioinformatics techniques. For the next part of this research project, we use transcriptomics and methylation data to identify biomarker panel for the stage classification of hepatocellular carcinoma. Here, we employ a TCGA dataset of 350 samples and elucidate 51 features that include 30 RNA transcripts and 21 methylation CpG sites using a number of data mining approaches. The name-based model classified early and the late-stage samples of hepatocellular carcinoma with an accuracy nearly 79%. For the third part of this research project, we used transcriptomics data to scrutinize the diagnostic biomarker panel for the cholangiocarcinoma, which is another major liver cancer malignancy. So in this study, we employed 9 datasets that containing 350 cholangiocarcinoma, 133 adjacent non-tumorous and 90 hepatocellular carcinoma samples to scrutinize cholangiocarcinoma specific transcriptomic signatures, implementing a number of machine learning techniques like extra tree, SVM, RVF, that is random forest, logistic regression, then LDA, etc. So in conclusion, we identify a universal platform independent 3-gene based biomarker that can predict HCC patients with high precision and also possess significant prognostic potential. Then we identified a 3-gene based cholangiocarcinoma specific transcriptomic signature that is capable to discriminate CCA samples from HCC and the normal with an accuracy nearly 90%. So at last, we identified a biomarker panel of 51 features that include 30 RNA transcripts and 21 methylation CpG sites to predict the hepatocellular carcinoma at an early stage. So during this program, I'm looking forward to being your mentor during this Omicslogic Data Science program and help all of you learn about data analysis, machine learning, and visualization in application to the large data set used in the biomedical research. In this program, I will be speaking about the role of big data plays in biomedical research that can generate a hypothesis that is then analyzed and used for the accuracy improvement and how this process is actually affecting clinical care. Now, I would like to hand over this session to Dr. Mohit Majumdar, who is the project mentor at Pine Biotech. So over to you, Dr. Mohit.
Thank you, Dr. Harpreet. Just let me share my screen. Hello, everyone. My name is Mohit Majumdar, and I'm one of the global representatives of Pine Biotech. My PhD was focused on developing a novel method to identify and predict the binding affinity of calcium binding sites. So as a result, I had to master data mining and become proficient at predictive modeling. In my current role, I am collaborating and working with academia and industry to provide support for bioinformatics education and research. I have 12 years of working experience and collaborating with pharma healthcare industry to solve high priority scientific questions related to biomedical data analysis and bioinformatics capacity building. So as earlier mentioned, there has been a remarkable shift over the last decade in the cost of generating molecular measurements. So it's just not genomes or DNA sequencing, but also transcriptomes, proteomes, and metabolomes. So most impacted by this change is the area of genomic medicine, where it is now possible to move from generating reference or population level data to producing data for individuals. So we are getting data from individuals. And concurrently, there have been also advances in the development of new algorithms and tools to integrate and interpret this data. So bioinformatics is playing a key role in all healthcare industry and especially in pharma industry. So the pharma industry is now considering data science and bioinformatics to be a very important part of their portfolio. If you see this latest survey, you will find out that AI and big data have the biggest impact. However, to understand this application and to be able to find information from this big data, it is important to learn from the analysis strategy that we have been discussing. And you saw the analysis strategy that Dr. Harpreet showed, and get experience working on real data sets that Ilya talked about. So together with our research collaborators from top universities, we have prepared projects for you that takes you through a project-based approach where we take data sets from high impact publications and use them for practical exercises. So it is, kind of difficult to imagine what the biomedical industry will look like in a few years' time, but it is certain that this surge in biological data flow will continue. And this increase in data will be because of many large innovative research projects, such as the International Human Cell Atlas Initiative 10, that aims to create comprehensive reference map of all human cells and also get data from devices like apps, wearables, and implantable technologies. So in this upcoming data science program, we will learn about analysis strategies and methods and getting hands-on experience on real data sets. And I'm really excited to be a part of this data science program, co-mentoring and working with students and researchers. And I'll be happy to help you learn the concepts and application of data science in biology. So with this, let me pass it on to Ms. Deepsha Biswas to take this forward. Thank you, everyone.
Thank you so much, Dr. Mohit. So I wanted to take you through some of the, okay, let me just share my screen first. Yes. Through the different program.
Okay, and as you're sharing your screen, maybe I can just answer a couple of questions that are coming up here in the chat.
Sure.
So the questions that you have could be divided up into two parts. The first part is technical. How can you actually get access to the program? What type of training will be included? What is the registration process? And all of these questions will be addressed right now by Deepsha as she walks us through the different resources and the process of registration. Now, the other questions are regarding bioinformatics and data science. So things like how to mine raw data, or maybe where to find the specific data sets that you can practice with, maybe the differences between different types of methods. So all of these questions come up typically and require some time to address. For example, it's not obvious how machine learning is different from statistical methods, or maybe different types of machine learning models differ in their prediction quality. So to understand the differences and the technical components of each one of these analyses and the different types of data, we'll have to go through a series of lectures. So when Deepsha shows the process of registration, she will also highlight the schedule of the upcoming programs and introduce you to a variety of resources that you'll have access to in a schedule, interactive sessions like today, as well as asynchronously. So you'll have access to those materials and you can use them during your own time on your own schedule. And the two work together to produce the Omicslogic program. So I hope that that clarifies it a little bit. And please go ahead, Deepsha.
Thank you so much, Ilya, again. So first of all, let's talk about the registration process. So here I am putting a link in the chat box. This is the link to register for this program, which is starting on the 12th of October. And it is a completely 1-month program, which will discuss about data science and machine learning for omics data. So as you scroll down through the page, and you can do that right with me as I'm speaking, you will see the different details of this program. And the next thing are the different session topics. So as you see that in the first session, which is again on 12th, we will be talking about the introduction to data types and properties. And on October 14th, we'll be talking about big data challenges and opportunities, and so on and so forth. So we will be starting at a beginner level and slowly advancing. So there is a lot of questions that we get. Who is this program for? And just to clarify it to everyone, and now that we know that all of you are coming from different levels, this program is suitable for undergraduate students who are just starting out and are interested in learning more about data science, as well as for faculty and researchers who are working in this domain. So as you scroll down more, you will find different resources, and you will find some testimonials as well from our previous participants, an overview of the different courses, and here you will meet our team. So how do you register here? You can click on Get Started, and you will be given 3 options to register. So the basic registration or the beginner registration will give you access to all of the resources that I will be showing you in a few minutes. But the intermediate membership will give you access to not only the basic resources, but also one-on-one mentor support. So if you have any problem during this entire 1 month period, you can actually get on a one-on-one Zoom call with our mentors and talk to them to solve your issues. So that is what the intermediate membership is about. And the advanced membership is for anybody who is doing a data science project. So you want to not only learn, but take it one step further. So you can opt for this membership, because this will allow you to go beyond this 1 month period, which is till November 4th, and do your data science program and your research project for another 30 more days. So if you have any questions regarding this, you can always reach out to me at marketing@pine.bio for more details. And now, once you have registered here and you chose any of these memberships that you would want to go forward with, you will get access to a profile like this. So here I have one profile from a previous participant. And as you can see that Grace here is from Sri Lanka, that I can understand because he wrote about it right here, and what he is doing, what he is working on, you can find right here. He has also added his social media link. So this profile serves as a really nice way to make a portfolio of yourself for not only the program participants, but also for anybody else who is a member of our growing community. They are able to interact and collaborate. And I'll be giving you some examples in a few more minutes. And as you see, Grace has a certificate. So as we mentioned, this particular webinar does not offer you a certificate, but you can definitely earn a certificate when you join one of these programs like this. So you see Grace has a certificate of excellence. So we give out a certificate of excellence or a certificate of completion, depending on how you're performing during a program. And also you can see an independent project that he developed along with his collaborators. So these are some of the opportunities. Now let's talk about the resources that you will have access to as part of this data science program. So the first resource you will have access to is this forum page or the organization page. So this page will serve as a classroom or as one place from where you can access all of the different courses, materials, even the discussions. We have a dedicated forum, which I'm going to be showing you. And also here is mentioned all of the different sessions for the upcoming days. So you will be able to see the timetable as well here. And then you have a forum. So what is the utility of the forum? The forum offers you the chance to see any news that is posted, any updates on the sessions, to interact with each other, and to ask questions. So say, for example, we have a session on the 12th of October, and after the session, you had quite a lot of questions. So where do you ask these questions? You post them right here on the forum by creating a topic that you can do from right here. And once you create a topic, it will be posted and we will be able to address that in the upcoming sessions or also reply back to you right here. So that is what the forum is about. Then you have access to a lot of different courses. So these are only an overview of the few courses that you have access to, but you have access to, as part of this program, to a lot more courses. So you see, we have these few courses which are dedicated: machine learning for biomedical data, and also the PCA biomedical data visualization in R, as well as projects. So these are the asynchronous materials that Ilya has been mentioning. Besides the live sessions, these courses and these projects will help you learn more about data science and explore it on your own at your own time and space. And moving on, regarding the independent projects, as you see, we have a lot of independent projects that were developed by our participants, our research fellows, and you can find all of them at this example. If you are interested in looking, you can go to each of these videos and see what these different participants have studied and what they are proposing in their independent research. So please go through this link if you're interested. We even have high school students who have been successful in utilizing the resources and developing their own independent projects, which they later presented at science fairs or won some competitions in. So please take a look at that. Here is an example of a poster presentation. You can also take a look at this. And to keep in touch with everybody beyond our own community, we have the Facebook and the LinkedIn group. So I had shared these links before in the chat. So I will be sharing them again. In LinkedIn, it is called Omicslogic Machine Learning, Biomedical Data Mining, and High Throughput Data Analysis, where we post about different activities, different research topics, as well as job news. So that is all from my end. Ilya, if you would like to go ahead and share more.
Okay, thank you very much, Peepsta. And I wanted to now address an important topic that comes up during these sessions, and the question of access. So the question of access is really about those of you that have minimal bioinformatics experience already that would like to benefit from learning about bioinformatics. And I want to introduce you to two different aspects of this problem. So the first problem is how not being an engineer and not having the capability to code from scratch, can you get started with analysis of big data? How can you mine publicly available big data? How can you generate those tables that you can then visualize and investigate for patterns? And even, how can you develop a sense of the logic behind the analysis that we will be discussing in the first part of this program? So for those of you that are still with us, I would like you to go to server.tbio.info. So this is the link. In this link, you will have a, thank you, Beepsa. So on this link, you will see the areas of analysis. So please let me know once you are on this site. Okay, you can just post yes or I'm here in the chat, and I will guide you on what you can do next. So we'll do a little demo and you'll be able to try it for yourself. So please let me know once you're here. Okay. We'll wait until at least a few people are on this page. So we'll wait until 5 people are on this link. Beepsa, if you could also post the first demo credential.
Yeah, I have posted both the credentials, so please try it with both.
Well, yeah, so if you could just use the first one, Beepsa, just maybe remove the second one for now, if you can. Okay, so for those of you that are on here, I see that maybe 3 people. Anyone else? Let's wait a little bit and maybe we'll get a couple more people to get to this link. Okay, so we've got 4. So one more person. Once you're there, we'll get started. All right, excellent. So now you can see all the way on the side, you will see a login link. So please press on login and use the first credential. So info@pine-biotech.com, and the password is 123 edutest. Okay, please let us know once you have logged in. This is important if you would like to follow along. Okay, thank you, Beep. So the username is info@pine-biotech.com. The password is 123 edutest. Once you log in, you will have access to a demo that illustrates how bioinformatics analysis pipelines are generated to produce structured data from raw sequencing reads. So let me give you a demonstration. Here on top, you see the different areas of analysis. Analysis of next generation sequencing data, mass spectroscopy, structural biology, as well as methods for data integration, analysis, and modeling. What we'll do today is we'll take a look at how to take raw sequencing read from next generation sequencing RNA-seq experiments and transform them into a table of gene expression that we'll use then to visualize using R. Okay, so first of all, you click on this down arrow and you can go to the cell line project demo. So you click on the cell line project and what you should see is this pipeline builder. Okay, so for those of you that see what I see right here, please put in number 1. Okay, so number 1, when you start your bioinformatics pipeline, is you need to be in the pipeline builder. Okay, so let's build a pipeline. First of all, we need to load the data, but as we load the data, you have to go through a series of steps to process this data. Each job produces a transformed data set that the next job will pick up and pass it on. So it's really like a pipeline that water flows in and you can generate different types of output. So here we have the main steps listed, which is pre-processing, mapping, and quantification. So we'll first click on start. Here you will find a number of algorithms that are publicly available and you can run them on your computer as well. But first, before running them, you need to learn about how they work, what type of inputs and outputs they produce, and what is the logic of using them to answer specific biological questions. So let's go to Top Hat 2. Top Hat 2 is actually a collection of different algorithms that includes multiple steps of data transformation and produces a number of different outputs. For example, it will produce a SAM file, a file that contains alignment of reads to the reference genome. So once you produce those reads and you align them to the reference genome, you can identify junctions between exons. And that gives you a sense of what kinds of transcripts are present in your data set. Once you've identified the transcripts, that information can be used to map on the transcriptome or map on transcripts and generate a table of expression that will look like this. Okay? So once we've generated the table of expression, we can complete our pipeline and you will get an output. So let me know how many of you have the output tables. So this should be a gene expression table. Okay. Now, these expression tables can be now studied using statistical analysis. But for statistical analysis, you need to have a hypothesis. You need to understand what kinds of groups do we have in our data? What kinds of conditions? How do we compare them to each other? So now please come with me to a different area that we'll also go through in this program, and we'll see how to use principal component analysis to be able to visualize what we have in this table of expression. Okay, please go with me to code.omicslogic.com. You actually have to register here as well. You cannot use the demo, but the registration is free. So you can register for free, and I'll let you register as we speak. But just to give you a sense of what you will have access to throughout the program, and even if you don't join the program, just if you have access to these tutorials, you will be able to work with these data sets and analyze them. So here we start with an introduction. How do we take the table of expression that we just generated on the TMyInfo platform, which is a gene count matrix, okay, and you can download it right here. And here we apply a method of what's called dimensionality reduction. So we reduce the complexity of this table that has thousands of genes and tens of samples. And once we do that, we can analyze our data for different groups. Now, before we jump into how the method works, let's see the coding part of it. So this is a code in R, and you see that its colors indicate different types of syntax. The first part is loading different libraries. So libraries are collections of functions that have inputs and outputs, just similar to what we just saw in the pipeline builder. But their objective is to simplify the process of writing code. So you don't have to write everything out. You just load somebody else's library, and you can call specific functions from that library, give it the correct inputs, and it will produce a result. So next we go into the input section. When we input a file, we intake everything that the file contains, the names of samples, the names of genes, and the numeric portion, which is actually what we will analyze. So once we have a record of what the input file is, we can read that table and specify where the header is, and importantly, separate the labels from the actual numeric data, which is the matrix of gene expression. Now, the methods of analysis that do require a lot of theoretical understanding, to practically apply them, you need to have minimal knowledge. You actually have to only know the specific call function, how to call them, and how to pass parameters. So here we have a call of a function called autoplot that will, the library called autoplot, a function called prcomp. So we'll actually do the analysis for us. And you can also extend it with different types of outputs. So let's see how this actually works. Okay, so here you get to the code part of it. And if you have the right data set loaded, which is right here, okay, you can just click on run, and it will produce an output right here in the console. Now, what we'll do in the context of this program is we'll try to apply this same method to a variety of different data sets and play around with the different settings so that we can improve upon these basic outputs. So for example, here you can see a 3D plot with the data set that we analyzed. You can also understand the model. So you can understand how much variance does each principal component contain. So to really go through all of these elements, we also have a full course, and that course explains how this method works with a lot more technical detail about every step of the way. So this is a little overview of some of the activities and resources that we will leverage to make this program effective, interactive, and enriching, because we will not only have interactive sessions where we talk and present the theoretical background of different projects, but you'll also have independent time to go through some of the materials on your own. Now, for those of you that are new to bioinformatics, I would like to recommend a different program for you to get started. So before jumping into data science, you might be thinking, this seems very advanced for me. I'm not ready to spend a lot of time and learn how to code from scratch and how to deal with these big data sets. So I recommend our Introduction to Bioinformatics for Biologists, a program that will simplify a lot of the things that we'll talk about and prepare you for next stages. For those of you that are more advanced and you already feel like you are ready to not only learn about these methods, but actually apply them to a long-term research project, and your objective is to publish, present a poster, work on your master's thesis, or incorporate bioinformatics in your PhD program, we encourage you to explore the Research Fellowship, which is an opportunity to learn about these methods in the context of your own independent research project. We meet once a week and discuss progress on your project, supporting you with assistance from our mentors, interactive community of interns that are all working on their projects. So you have a way to collaborate, get feedback from experts, and answer any kind of technical challenges that you need to overcome. So again, thank you for joining today's webinar. I hope that these resources were explained clearly, and thank you to Beepsa that did an excellent job going through the technical details and the logistics. You'll hear from us again by email. We'll follow up and see if you need any assistance with anything that we presented today. I do want to mention that in all of the resources that we shared, there's a free level. So you don't have to pay anything to get started. And what I would recommend is to get started with our course on Introduction to Bioinformatics. So the Introduction to Bioinformatics is a course that provides you with a broad overview of the way bioinformatics is used in medical research, basic research, agriculture, biotechnology, and the pharmaceutical industry. And it's a great introduction to a lot of these topics, as well as a survey that you can fill out to let us know more about you and help us design a personalized plan for you to be able to benefit from the resources that we have. So again, you can register here for free, create your profile, become a part of our community, and start learning for free. And these courses have certificates that you get upon completion, even the free courses do. And you can explore very affordable ways to continue and expand your knowledge as you go through. Now, also on this platform, the coding platform, there are a number of free courses here as well. And there's a variety of different types of courses here, including getting started with bioinformatics on how to process sequences, how to deal with sequence similarity, how to get started with data science in R and in Python, how to deal with genomic data, metagenomic data, and transcriptomic data. And this list will continue to grow as you become a part of this program. Thank you again, and thank you, Mohit, for presenting, as well as Harpreet for presenting about your research project. We'll be looking forward to seeing all of you in the program when it starts on October 12th.